Welcome to Between the Signals, a podcast brought to you by the UK Dementia Research Institute at King's College London. As our population gets older, dementia has become a major health challenge. If nothing changes, half of us could be affected by dementia in some way, either facing it ourselves, caring for a loved one, or both. What is a neurodegenerative condition? How might these conditions affect us and our future as we age? These are exactly the kinds of topics that we're breaking down on this podcast, and we hope you enjoy.
Hello everybody, and welcome back to another podcast episode of Between the Signals. My name is Dr Sarah Mizielinska, and I am a group leader at the UK Dementia Research Institute. And today we have the enormous pleasure of having Professor Chris Shaw here with us to talk about his experiences, his amazing discoveries in the genetics of both motor neuron disease called MND, but also known as amyotrophic lateral sclerosis, ALS, and also how he's taken these discoveries through to have a spin-out company of his own and actually get therapeutics to people affected by neurodegenerative conditions. So welcome, Chris. Great to have you.
Nice to see you. Thanks, Sarah.
And so we want to take you right back to the beginning and how you came into research and how this journey began.
Right, well, I should start in New Zealand because that's where I'm from. I trained in medicine and I wanted to become a hospital doctor, a clinician, a physician. And so I do a variety of different subspecialties, but neurology for me was the most challenging. There were no treatments for most conditions. We didn't really understand what was going on in the brain. In New Zealand at that time, we didn't even have MRI scanners, right? So it seemed to me to be the area in which there was the greatest need for improvement and advances. And I started doing some, sort of research in the weekends and at nights on a condition called multiple sclerosis, which is an inflammatory condition of the brain. And we were looking at how the inflammatory system was being switched on in this condition. And I managed to write some papers and got a Wellcome Trust Fellowship to come to Cambridge in the UK. And for me, that was transformative. But my first encounter with people with motor neuron disease was in New Zealand, and it was very depressing because obviously you're giving people a terminal diagnosis, and we knew that the disease was progressing pretty fast, but most people don't know what's coming. There was very little known about motor neuron disease. But when I came to the United Kingdom and I was working in a small clinic, I met my first patients who had a familial form of it. That is that they'd inherit it from a parent. And they knew exactly what was coming.
Yeah.
And talking to them and hearing about the burden that they'd carried all their lives, every time they tripped on a pavement or had difficulty opening a door, they thought this was the beginning of the disease. Not only that, they were two women in their 50s, and they knew that if they carried the gene, that they would have then had the chance to pass it on to their children. And that's the sort of, the impact of that on them really changed my focus to then spend the rest of my life trying to work and understand this disease process and develop some therapies.
You started understanding these familial cases. So where did that take you to in your research journey? What did you come to Cambridge to do?
Yeah, so I went to Cambridge to sort of look at how nerve cells interact to be able to help the brain recover from multiple sclerosis. But I learned a lot about how nerve cells and the supporting glial cells work in systems. We were able to grow them. We were able to look at the impact of various genes. But it was this familial motor neuron disease that really attracted my attention about going forward because at that stage, the first gene was discovered. It was called SOD1. Okay. And indeed, both these women carried SOD1 mutations. Right. And that led to me to work with somebody at King's called Professor Nigel Lee, who was an international expert in this area, and he helped us identify these gene mutations, and then we started to study the disease process in these people. So that's how I got started. He offered me a job at King's, and I've been here for 30 years.
So yes, you have, been instrumental in the discovery of many genes and genetic risk factors for motor neuron disease. And can you tell us a little bit about how that process, how does that occur? How do you find these genetic risk factors?
Well, it was extremely difficult. I was employed between the neurology department and the genetics department, and the professor of genetics said, motor neuron disease, that's not genetic. Anyway, it's because very few people perceived it to be genetic. And the other challenge was, in traditional genetic linkage studies, you tried to discover a gene that was running through multiple generations in very large families. In motor neuron disease, everybody who's had the disease in previous generations is dead. Right. And so we had to try and recreate missing people from these families by genotyping the spouse and the children and to see what genes the missing spouse would have had. And that's a very slow and difficult process. So it was a pretty imprecise science, which took us a long time to discover more genes. And the first really big gene, an important gene that we discovered was TDP43.
You mentioned genotyping. So what's the link between genotyping and genetics?
So most people know about DNA and sequencing. So there are various signals that we can see tracking along different chromosomes. And the way we genotype people to do linkage is to see which, if you like, markers people are carrying in their different chromosomes. And we can track down which chromosomes are being shared amongst all the people that carry the disease and not shared on those who aren't affected.
And so with all these genetic understandings, how do these help tell us about the kind of biological underpinnings of these conditions?
Well, it's really interesting. So many of these genes actually lead to defects in the same part of the cellular machinery.
Right.
And a particularly important part that I'd like to focus on is the protein recycling machinery. That seems to be hit by a number of different genes, which can all cause motor neuron disease or frontotemporal dementia.
So you mentioned frontotemporal dementia kind of in line with motor neuron disease. Why do you mention these two together?
Well, they seem to be related, both in terms of the genetics, but also in terms of their pathology. So we know that when we look into the brains and spinal cords of these patients, the same sorts of proteins and the same sorts of genes are being affected. So we now think of them as being a spectrum of disease. And some people have more frontotemporal dementia, where the frontal lobes and the temporal lobes take the burden of disease. And other people, sometimes even in the same family, it's the spinal motor neurons that are most affected.
And so can single individuals have both of these diseases?
Some very unfortunate people will be affected by both conditions, neither of which are great.
But it's also then understanding the genetics and developing therapeutics for these means that we could develop therapeutics for both, right?
That's a really exciting opportunity. That this isn't just going to be about one single gene in one small population of patients, but we could generalize that across that. And TDP43 is very interesting because we know it's important for frontotemporal dementia, motor neuron disease, but about 40% of people with Alzheimer's disease and Parkinson's also have TDP43 accumulating, and we know in those individuals it's also contributing to disease and symptoms.
So speaking about therapeutics, not only are you a clinician and a researcher, both very successful ones, you're also now an entrepreneur and have your own spin-out company, Aviado Bio, developing and testing gene therapies for neurodegenerative conditions.
I don't think of myself as an entrepreneur, really. I think of myself as a monopreneur. This is the only company I wanted to spin out, and I'm not sure I even wanted to. It was kind of an accidental event. Yes, so I've been working on discovering diseases and mechanisms for a very long time. We generated a lot of different discoveries. We generated cells and mice that carry these genes that people were using around the world. We made that available to everybody. But actually, the therapies weren't coming through. There was very little progress. And so we decided to do this ourselves. I was very fortunate in working with some people who had experience of gene therapy before, and together we formed the spin-out company. So I wouldn't say it was my motivation, but in the end, we felt it wasn't just that we should go on making more discoveries about an explanation about disease mechanism. We should actually try and develop some therapies.
So, and how has that been then? How has that journey to really developing a successful company been?
So the most important thing is to work with people who have that expertise. And so I was extremely fortunate in being able to recruit really good people who had experience and who were able to develop therapies and bring them to the clinic. And so the success is theirs, much more than mine. So we started off with a therapy for frontotemporal dementia due to progranulin, and we went about it in a different way from what most other people were doing.
Okay. And how, you mentioned it's different. So how is it different?
Well, there's something special about progranulin. These patients who have mutations are deficient in progranulin. And so because they don't have enough progranulin, the cells that are involved in the protein recycling tend to fail. And the the mechanism within those cells starts to fail and those cells degenerate. And so the idea was that we would give progranulin back. And the reason that we chose progranulin is we knew that it was secreted. And so that if we get into part of the brain and get it delivered around the brain, then we might be able to have a therapy for the whole brain.
So is that why you chose progranulin over other ones that you've mentioned, like SOD1 and TDP43, these other genes that play a major role?
So the first thing was that it was a supplement, and that's easier to do than silencing these genes. And the fact that it's secreted means you could go from one cell to another.
Okay, and so let's follow up on that. So one cell to another. So what are we talking about here?
So we wanted to make this the best distributed protein in the brain. And so we looked at a part of the brain that was best connected to other parts. And we chose the thalamus, which is this small structure in the center of the brain, which takes sensory information from the rest of the body and delivers that information to the brain and the cortex. And so we asked the thalamus to be the factory to make the progranulin. And we asked it to then distribute it along axons, right, and then have it secreted at synapses and being then taken up by other cells in the cortex. And much to our surprise, this did actually work, firstly in mice, and then in sheep, and then in monkeys.
Okay, so I'm just going to clarify for the audience that brain cells, nerve cells, neurons is all the same word for the same thing. They have these projections that we describe a bit like a tree, the branches of a tree, and some of these are axons, and these are long projections that connect one cell to another cell. And at the end of these is a synapse, and that really is the connection between two cells. So Chris is talking about his therapeutic traveling along from one cell and being transported across the synapse to another cell and therefore being distributed through the nervous system, right?
Correct.
And so what type of therapeutics are these, Chris? Like, how are we supplementing this progranulin?
Well, the way we delivered the gene was to package it in a virus.
Oh.
Which sounds rather scary, but actually what we're asking the virus to do is just deliver our gene. Normally viruses have their own genes so they can replicate and cause infection. That's what they're out there to do. What we're using is the shell of the virus, and we're putting inside the gene that we want expressed, not the viral genes.
And so are there no dangers of using a virus like this?
Yeah, so this is a very small virus. People do get exposed to this virus, but it doesn't really cause any systemic illness. So that people don't get sick with this virus.
Right, okay.
And we know it's been given to thousands of people quite safely. We have to worry about dose and all the sorts of other things, but actually it's a pretty safe sort of vehicle to deliver the genes to the right cells and do the right thing.
And so what's the benefit of having like a viral delivery or a genetic delivery of a therapeutic versus having the traditional kind of small molecule chemicals that are, like paracetamol and things?
Yeah. So a lot of drugs don't get into the brain.
Right.
The brain has a barrier, so things in the bloodstream don't get across, and that's a good idea because you don't want most viruses and bacteria getting into the brain. It's a very delicate organ. So some small molecules do get across the blood-brain barrier, but actually they have an effect on every single cell. The nice thing about using a virus to deliver your gene is you can put that gene under certain regulatory control, so it's only switched on in certain sorts of cells or in certain sorts of conditions.
Right. And so I think you mentioned before, as well as having those regulatory elements, that you also directly inject that into the brains of people. Is that what we're talking about here?
It is. And again, that sounds quite scary. We're going to inject a virus into your brain to give you a gene that you're missing. But actually, that has been done in other conditions and been shown to be very safe. The part of the brain we're going to, is regularly the site for the delivery of a probe to treat tremor and Parkinson's disease. We use deep brain stimulation. And this is just a virus that we infuse into that brain, and we take the catheter out, and you're left with the virus in the brain. We know from our animal studies that actually it's a pretty safe thing to do, and you get long-term expression in the nerve cells. And the nice thing about that is that one treatment could be a treatment for life.
Right.
Because the gene keeps being expressed over time.
Okay, so that's really beneficial over having to keep taking a medicine over and over and again.
It's a one-time therapy.
And so it sounds like that's quite a specialist kind of procedure. So this is not something you're going to go to your GP for, is it?
No, and that's a challenge. We've gone to centres in the world that are experienced in delivering gene therapies to the brain. It has to be done under an MRI scanner, so we know exactly where the infusion's going, and we can see if there are any problems during the surgery. So the first phase 1 and 2 study is all about safety. Can we do this safely? And then we can answer, are we actually getting a therapy made in the person? And then the third question is it effective in treating symptoms?
You've made incredible progress in your company, taking a gene therapy into humans beginning last year. And so how does that feel?
Scary at first, but we're now on to our 3rd cohort of patients, so we're pretty confident about the safety of the procedure. And now we're trying to see a signal as to whether we've actually delivered what we thought we were delivering, and whether subsequently we'll find out whether it's been effective in terms of reversing the disease process or slowing down progression.
And so we've talked about your incredible efforts targeting progranulin, but so what about the other causes that we talked about before, TDP-43 and SOD and other ones? Like where are we as a field and your company progressing targeting those things?
So we chose progranulin because it was secreted, right?
Yes.
But it's actually a very small number of people who carry these mutations that would benefit from that particular therapy in the first instance. For many of the other genes that cause frontotemporal dementia and motor neuron disease, actually it's a toxic protein that's being made.
Right.
So you need to switch that gene off.
Yeah.
That's much more difficult.
Okay.
And you need then to deliver your therapy to every single cell you want to save.
Right.
So rather than asking the factory
at
the center of the brain to go and distribute it, we now have to get our virus all around the brain, and that is difficult. What I would say is that there are advances in terms of the virus capsule that are being made so that it can get across the blood-brain barrier. And so that's a really exciting development, because if we can get it across the blood-brain barrier safely, then we could get it to deliver almost every single cell in the brain. And indeed, there are now capsids out there that are doing that, and we are working with them.
So we've talked about targeting these toxic proteins, so from these genetics that we mentioned earlier, like TDP-43 and SOD1. Do we need to target all of these different genetics? Is that the plan? Like we go after all of these big genetic factors that...
So that would be pretty challenging, and I think there may be individual strategies, but what I have been focusing on in the company is to look at where they come together. I kind of think of this as like different tributaries to a main stream.
Okay.
And rather than tackling every single tributary individually, I'm going to look at the pathways where they come together, in particular around TDP-43 protein accumulation. And I think we have got some important genetic clues there as well. And if you can dam that river, then perhaps you can delay or slow or even arrest the disease process downstream.
So do you think is it better to target downstream, or do we need to target upstream as well, or what are we talking about here?
I think a bit of both. We talk about perhaps combination therapies in the future. And so there may be targets that are for an individual with a particular gene variant. You might target some element there, but you also want to target the mainstream as well.
Of course.
And it won't just be gene therapy. There'll be other small molecules that will make a contribution, and this is just one particular approach that we're tackling.
So it sounds like we're making amazing advances in gene therapies and targeting genetics and downstream kind of these places, as you say, that fill into this stream. So what's your future perspective on where are we going to make the best impact to people with these conditions?
Okay, so I think if we can show that these gene therapies are effective, that's a great start.
Yep.
But one of the problems with a one-time therapy is you can't change the dose.
Right.
And so, that's the dose you're going to get for the rest of your life. What we are looking at now is ways of tuning that therapy. So we can turn it up or turn it down. And there are small molecules that cross into the brain that are able to trigger the therapies. The other thing to think about would be, could we put inside our therapy a trigger when that cell becomes sick?
Right.
So it's able to detect a change in the cell's biology that suggests it's beginning to struggle, and you switch on your therapy in those cells. And then we really could treat people who are at risk, who carry these genes, who are not affected, and the therapy will only start up in the tissues that are really being affected at the time they need it.
So this is not just about making more therapeutics, it's also making our therapeutics smarter as well.
Exactly.
Yes. And so how do people with these neurodegenerative conditions feed into this research? Like, what role do they play in the development of these therapies?
So they're at every single stage of the process. The reason I started working in this area was because the need was so great, and these people were facing a really bleak future. And although they knew that I was really unlikely to be able to help them personally, what we were hoping to do is to change our understanding of disease and develop therapies that would help their children. Yeah. And we're still doing that. And obviously I've been doing this a long time, so I'm now seeing the children to come through. And sometimes we do have a therapy that really has changed the options going forward, and I think for SOD1 that's definitely true. There's a therapy that really can work and is very powerful, but for the majority of people we're still waiting for that next breakthrough.
But there is hope.
There is hope, and we now have the tools to really tackle these in a serious fashion in a major way.
And you've made an enormous contribution to this, Chris, over your career.
It's very kind, but it's a huge team effort. I mean, the company is now 60 people, all experts in gene therapy. I just lit the fire. They've been fueling it.
It's always a team effort in science, right?
Yeah, always.
Always. Thank you so much, Chris, for coming here today and sharing really how you got into research, your discovery of so many genes across motor neuron disease and also other diseases, and then really taking these kind of fundamental discoveries all the way through to developing treatments and really kind of bringing those to people with neurodegenerative conditions. Thank you so much.
Pleasure.